The nucleus is made of a nuclear envelope, who is a distinctive sign of a eukaryotic cell. His major role is separating the inside nucleus from cytoplasm. Also, the nuclear envelope contains nuclear pores, who allows the exchange between inside nucleus and outside. Hi guys, and welcome back to the next chapter from nucleus course. Like you see at start, today's lesson it's about nuclear envelope and pores. It's gonna be very detailed, so let's get started. First of all, let's start with structure. Is a membrane surrounding the nucleus from outside and separated her from the cytoplasm, and is very important. He's a distinctive mark for the eukaryotic cells, and we don't find them in prokaryotic cells. The nuclear envelope made up for two fine membranes. One of them the outer membrane and is faced to the cytoplasm, and the other inner membrane who face the interior of nucleus. The exterior membrane is united with the rough reticulum endoplasmic in certain places. The inner and outer membranes bind together in places and separate in other places, forming as gaps. Those gaps called the nuclear pores. The nuclear pores are very complex structure that facilitating communication between the nucleus and cytoplasm by directing the nucleocytoplasmic exchanges. The small molecule and ions diffuse freely, while big molecules are transporting via active transport or mediated protein. We see it how in this video. Nuclear pores have a diameter and thickness of 180 nanometer and 15 nanometer depending on the cell type. Now let's go to the structure of the nuclear pores. Every nuclear pores are surrounded by a cylindrical complex. Both centric membrane of the nuclear envelope fuse with multi-protein complex that manifest by three disparate protein termed NUP or nucleoporine. The nuclear pore complex possesses octagon symmetry. The structure of the nuclear pore complex comprises the following key elements. First of all, the cytoplasmic ring is united with about 8 filament cytoplasmic is working a major role in importing molecule inside the nucleus. The second ring called nuclear ring because he's facing the inside of nucleus and is binding with nuclear basket, which is the key element for exporting molecule to cytoplasm. Between the cytoplasm ring and nuclear ring is the central channel. Some nucleoporine like phenylglycine repeats account for formation of the central channel. The nucleoporine of the central channel function as a selective barrier which only allows the import and export of large biomolecules across the biliary nuclear envelope, of course. Now let's go and talk about the nuclear pores transport. Like we said a while ago, that nuclear pores allow the communication between nucleus and cytoplasm. There are small molecules and ions which travel without any help, and we call it free diffuse. And big molecules like protein are used in active transport or mediated by other proteins. We're divided into types. Important, we found them in cytoplasm. They import molecule inside the nucleus, and the second transport protein is exportin, which export molecule outside the nucleus to the cytoplasm. The importin we found him in cytoplasm, and the exportin we find him in the nucleoplasm. So let's start with the importing technique. Let's say we have a protein to import inside the nucleus. Only subserve protein can import to the nucleus. They mark it by a special sequence of amino acids called nuclear localization signal, and they consist of one or two short segments with basic amino acid. This segment recognized by the importin receptor and carry the cargo protein to the nucleus. In the way to nucleus important bind to the cytoplasm filament allow the cargo to go through the other sequential nuclear proteins until he get into the nucleus. On the other side, the complex is disrupted by protein called RAN and is carrying molecule of GTP. This protein bind to the important and change his structure, making him release the cargo. The protein complex of RAN important re-exported through the nuclear pores to the cytoplasm, and there we found another protein called RAN gap, and he bind to the complex RAN important and stimulate RAN protein to hydrolyze his GTP to become a GTP. This action make the RAN release the important receptor. The RAN GTP can never bind to the complex importing protein. In order to import the RAN GTP inside the nucleus, a help is required by protein called anti-Phe2. He binds to the RAN GTP. The importing then import him inside through the nuclear pores, then release him. Now we need the rank GTP to become a RAN GTP again, and that's what's going to happen by the help of protein called RAN GAF. Bind to the RAN GTP and stimulate him to release GTP and pick up the GTP. And the RAN GTP is functional again, and then he can bind to importing and trigger him to release the cargo. Now let's go and talk about export from the nucleus to cytoplasm. Proteins are targeted to export from the nucleus are marked by specific sequence of amino acids called nuclear export signal. They are recognized by receptor called exportin, and they bind with the RAN GTP who form a stable complex, the protein. The protein complex go through the nuclear pores to the cytoplasm. The RAN gap bind to the complex and stimulate RAN GTP to hydrolyze it to the RAN GTP and make him to release the cargo to cytoplasm. Exportin and RAN GTP are recycled by the protein anti-F2 protein and transporting them inside the nucleus again. That's it guys, the end of chapter 2. I see you in the next chapter. Don't forget to subscribe. I leave you with the quiz of chapter 1. I see you next.